Life Sciences Video Lecture 5, pH and Buffer Systems. So let's take a look at our objectives here. Today in this lecture, we are going to define organic and inorganic molecules and give examples of each, and define acid, base, salt, and the pH scale, and describe the use of buffer systems in the body. So these are our last two objectives that we're covering before the first exam. So basically what we're looking at here is part of what we're doing is looking at the difference between organic chemistry and inorganic chemistry, and also then how that shows up in the study of the body. From here, the next step then is to go more deeply into looking at the inorganic aspect of chemistry within the body, and then from there we'll look at organic chemistry within the body. Okay, so if we look at our notes and we look here at our at the slides, the first thing we have here is the term organic. The term organic refers to living matter. So like physics, the field of chemistry has grown over time, and as we discover more, essentially we eventually discover so much within a field that we have to kind of create offshoots of that field, so that because we know too much for it to just be one field. And we saw that in physics, where we went from physics was all there was, and then 3 more fields of physics needed to be developed to account for everything we were able to observe as we began to expand our awareness of the universe. Similarly in chemistry, the more we study chemistry, the more we understand that there are some very significantly different types of molecules that we can study and look at. And so we divide the categories of chemistry into 2 main groups. One is organic chemistry and the other is inorganic chemistry. So the basic difference is organic chemistry organic chemistry is more related to biology and comes up more in the study of cells and what cells are doing with their large molecules. Whereas inorganic chemistry can be applied to the body, and we're going to take a look at inorganic chemistry throughout the body, but it is also applied everywhere else as well. So we're only going to find organic chemistry within living systems, whereas inorganic chemistry are supportive of living systems. So we find them everywhere, but they're not. They don't necessarily involve a living system. So let's take a look at some of our definitions here. So we've already looked at organic. Organic refers to living matter. So, in today's world, if you go to the grocery store, organic means it was grown without pesticides. But in the world of chemistry, organic refers to living matter. So something with organs. It has organs, it's organic. So that's living matter. So organic molecules, when we talk about molecules as being organic or inorganic, organic molecules are large carbon-based molecules found only in living systems. So let's talk about those a little bit more. Let me see here. So here are some examples of organic molecules. But here, there are a few characteristics that differentiate organic molecules from inorganic molecules. One is that organic molecules are carbon-based. So if we look at these examples, you can look at these and go, I don't see very much carbon there at all. Well, that's because these molecules are so large. So for instance, look at the one in the upper right, which is stearic acid, which is a type of fatty acid. That actually has so many carbons and so many hydrogens that we can't even show them all. We can, but it becomes quickly very unwieldy. And so we depict the molecule in this shorthand kind of way. And so with this shorthand, what we're looking at is each of these peaks and valleys in the zigzag. Every time there's a corner, there's a carbon molecule there. And so each of those lines represents a covalent bond between 2 neighboring carbon atoms. The other thing that is implied, the way that this is depicted, is that for each of these carbon atoms, there is a single covalent bond between them. And we know, do we know? We haven't talked about this too much. Let me do a little sidetrack here. Carbon atoms have the ability to form 4 covalent bonds. That's what makes them such a good structural foundation for large organic molecules. So if we look at a carbon atom, we see that the carbon has its atomic number is 6, which tells us there are 6 protons in the nucleus and 6 electrons. So 2 electrons in the first shell, 4 electrons in the second shell. We know that the second shell can hold up to 8. So that means that carbon, well, is its valence shell half empty or half full? This sort of, the question, right? Is it half empty or is it half full? Carbon, I think, likes to look at itself as half full because it does not tend to get rid of those electrons. It tends to share electrons. So it wants to fill up that outer shell. And so it has the potential to form 4 covalent bonds, and it does that often. And those 4 covalent bonds, you can kind of think of carbon as being like the ultimate Lego or something. It's like the perfect building block that you can put together and build lots and lots of things with it because you can attach 4 atoms to it with covalent bonds, or fewer if they're double covalent bonds or triple covalent bonds. But very often carbon will form 4 covalent bonds, or it will form a double covalent bond and then 2 single covalent bonds. That's typically what we see with carbon. So anyway, carbon is this great foundational building structure for these very large organic molecules. So if we look at stearic acid again, this fatty acid, so each of those peaks and valleys, at each of those corners, we have a carbon atom. Each of those lines of the zigzag represents a single covalent bond between 2 neighboring carbon atoms. And that it is a single covalent bond implies that each of those carbons also has a single covalent bond between 2 hydrogen itself and 2 other hydrogen atoms. So we, just because of the way it's depicted, we just know this. Because that's how organic molecules are drawn. Because if we were to draw in all of those carbons and all of those hydrogens and all of those bonds, there'd be just so much here to look at. So this is sort of a shorthand version of drawing an organic molecule. The one below it is cholesterol. And again, we see a couple of carbons here, but we don't see a ton of them. There is one little zigzag at the top of the cholesterol molecule, and that is like stearic acid, where it's carbons at each of those corners. But then a lot of cholesterol is these 4 rings that you see that's more on the left-hand side of the molecule. And those 4 rings are essentially rings of carbon. So again, at each of those corners, we have a carbon molecule, and if it has a single covalent bond between it and its neighbor, then it has 2 hydrogen molecules bonded, or 2 hydrogen atoms bonded to it as well. You can see one place where there's a double bond down at the bottom of the cholesterol molecule. So way at the bottom of the molecule, there are 2 hexagon shapes side by side. The hexagon that's on the right has 2 lines on one of its sides. That indicates a double covalent bond. So that means that those carbons between that bond have, they're sharing 2 sets of electrons between themselves. And so then, then that will impact the number of hydrogens that are or are not attached to those carbons. But anyway, so looking at these starts to give you a little bit of a sense of organic molecules. Let's see what else is in our notes here that we should talk about. So organic molecules, like we've seen, have a lot of carbon in them, and they have even more hydrogen in them. But oftentimes when we look at at the depiction of it, like the way we're looking at it on the slide, we don't necessarily see all those C's and H's the way we would expect to. But that's implied here, that there's a lot of carbon and a lot of hydrogen. We also have a fair amount of oxygen, which we can see here and there, especially like if you look at glucose. You can see a number of oxygen molecules. So all of those O's there are oxygen molecules. And you can see some oxygen molecules below glucose in that molecule, which is lysine, which is one of our amino acids. So we see oxygen fairly frequently in organic molecules, we also see nitrogen and phosphorus especially in like DNA and RNA. So the size of organic molecules ranges from, like, for instance, glucose is one of the smaller organic molecules that we'll find that has like 6 carbons in it. But organic molecules can range all the way up to hundreds of atoms to thousands to even millions of atoms, like in the case of DNA. Okay, so we primarily see covalent bonds in our organic molecules, but there are also hydrogen bonds in these molecules, and the hydrogen bonds are the bonds that actually, so because these molecules are so large, we can't think of them as sort of being one or two dimensional. We need to think of them as being 3-dimensional. So they have bends and curves and twists to them that give them 3 dimensions, but those bends and curves and twists need to be held in place so that the molecule doesn't fall apart if it's moving around in a solution or whatever. And those are held in place with hydrogen bonds. So recall that hydrogen bonds are a bond between a slightly negative and a slightly positive part of either 2 different molecules or 2 different parts of the same molecule. And so that helps to create that structure that we need to make our large organic molecules work. All right, so organic molecules in the body, what these guys are doing is they are our molecules of storing and releasing energy. They are the molecules of the structure of the body, and they are molecules of storage and transference of energy or of information throughout the body. So some examples of organic molecules are DNA and RNA. Those are our nucleases. So our DNA, where our genetic material is stored, and RNA, which is a temporary molecule that stores genetic information. Those are organic molecules. ATP, which is our energy exchange molecule in the body, and we'll take a closer look at all of these when we look at organic molecules. And proteins and lipids and carbohydrates are all, and we think of these as nutrients, and they are. So these are all things that we can take in through the food we eat, but we can also build any of these, proteins, lipids, and carbohydrates. And ATP, RNA, and DNA, we build all of these. And so, of course, it would take quite an anabolic reaction to build these large structures, but that is what our cells do. And it would take quite a lot of catabolic reactions to break these things down, and that is also what we do. If we need to break down old cells, we'll break down all this stuff. Or if we're breaking down cells of food that we've eaten, there will be proteins and lipids and carbohydrates and DNA and RNA all in there, and we'll break those down. And so that's what a lot of what's happening in the digestive system. Okay, so organic molecules are large carbon-based molecules that are doing kind of the big functions of living systems. Now, by big functions, we mean, these are still microscopic, they're happening inside of cells, but this is, energy, making energy, storing energy, this is building the body, this is communicating throughout the body. All of those things happen with organic molecules. Inorganic molecules, to contrast them, are small molecules. So they might have some carbon in them, like carbon dioxide, for instance, is an inorganic molecule that has carbon in it, but these are teeny tiny in comparison to the organic molecule. So inorganic molecules might be only 2 atoms or 4 atoms, or, they're probably not going to be much bigger than, not much bigger than 12 atoms. That would be a fairly sizable inorganic molecule. But the presence of carbon is not the defining feature of inorganic molecules. I mean, we could almost say the absence of carbon is more of a defining feature, but there are inorganic molecules that have carbon in them. So inorganic molecules are found in living systems. They're also found outside of living systems. They create environments. So they create the environment of our air. They create the environment of the fluids of our body. They create the environment of the ocean. They create the environment. They're what rocks are made out of. They are the earth and all of the environment that we live in on a macro scale and on a micro scale. And so these are molecules that support life. They're like the backdrop in a sense or they're the scenery or they're the environment. That all of the rest of these bigger organic functions are happening within. And if we don't have the right balance of our inorganic molecules, then reactions can't happen. Chemical reactions can't happen in the body the way we need them to happen. So inorganic molecules are small non-carbon-based molecules that create the environment for organic molecules and for chemical reactions. So let me just see. So these inorganic molecules, some of them that we have in the body. Do I have examples here? Well, water is an example that I have here. So the foundation of the internal environment of the body is water. Water is kind of our foundational inorganic molecule, and then we can add things to that water. Water is the medium within which chemical reactions happen. It's the medium that's used to transport things throughout our body. And so, water is extremely foundational for life. It is an inorganic molecule. Now, what's in that water also then is going to allow or disallow chemical reactions that need to happen in that water. So the body needs its internal environment to be kept within strict parameters. And so we have to control what is in that water so that reactions can happen properly. So some of the elements that we're going to see that make up these inorganic molecules are things like sodium, potassium, calcium, magnesium. In the body, very often we're going to hear those guys referred to as electrolytes. These are molecules, and they might just be in an atom form, but they often hang out in their ionic form. And that's true of a lot of inorganic molecules, but not all. But in the body, the electrolytes definitely do hang out in their ionic form. And so these are the substances that allow us to, for instance, conduct electricity through our body, which is how we get our nerves or how our nerves communicate, and how we get our muscles to contract, how we get our heart to beat, and all of those rather foundational important functions that happen. The pH, we're going to talk about pH here next, but the pH is of the fluids of the body is kept in balance by these inorganic molecules. Some other inorganic molecules we find in the body, or I could say, and again, some of these might just be atoms. They might just be, the element, or they might be parts of small molecules. So some examples are zinc, iron, manganese, copper. These are all things like, if we're taking those things in through our body, we're not going to call them macronutrients the way we'll call our proteins and carbohydrates and lipids. We'll call those macronutrients. These guys are minerals, so vitamins and minerals are things that we need. Vitamins are small molecules. Often they're inorganic, or they're a combination of a small organic molecule with some inorganic component to it. But minerals are minerals. Iron is a mineral, and that's something that we need in the body to make certain things work the way that they do. So we do see a combination of organic and inorganic matter together. So like hemoglobin, for instance, is a large protein that allows us to carry oxygen throughout on our red blood cells. Hemoglobin, a key component of hemoglobin is iron. So iron is inorganic. The oxygen that it carries is inorganic, but the molecule itself, the protein, that big structure that's called hemoglobin, that is an organic molecule. Okay, so our gases in our body are also inorganic. So oxygen, carbon dioxide, those are things we hear about a lot. Those are inorganic molecules. And the acids and bases and salts in our body are also inorganic molecules. So our focus here today in this lecture is on the internal environment of the body. So we're starting to kind of lay this foundation about what is the interior of the body and what is that environment and what support goes into it to make it what it needs to be in order to support the life of the body. So let's then go from here and talk about pH. So that takes us actually to the next topic, which is water. So I mentioned that water is the foundation of the internal environment of the body. Now, water, when we've looked at it, we could think of water as being made up of one oxygen of atom and two oxygen of atom, one oxygen atom and two hydrogen atoms covalently bonded together. Okay, so that is an accurate depiction of water. That's what we're looking at here. We can see on the bottom we have one oxygen atom, and then on top we have two hydrogen atoms. They're sharing electrons, therefore they are covalently bonded. So yes, that is water. However, we could also think of water in ionic terms. Okay, so if we think of water as being made up of ions, what ions is it made up of? Well, it would be made up of a hydrogen ion. So let's remember that a hydrogen ion, so on the left here, what we're looking at is a hydrogen atom in its natural state. So it has its one proton in the nucleus and its one electron orbiting around it in that first shell. On the right, what we see is the hydrogen ion, which is a cation because it has a positive charge, because what has happened here to turn this into an ion is that electron gets lost, which means that whole shell that can hold the electrons is gone, and all we're left is that one proton in the nucleus. So remember then that a hydrogen ion is basically just a proton. So anyway, in being so, it is a cation because it has a positive charge. So this is a hydrogen ion. So that's one ion that could be a part of water if we were to think of water as being ionically bonded rather than covalently bonded. And basically, water can go either way. It usually does have covalent bonds that are holding it together. But if you've heard of ionized water, that's water where the bonds are slightly different and is actually ionic bonds that are holding the atoms together. So the other ion that would make up water would be the hydroxide ion. So here's a depiction of a hydroxide ion. So what hydroxide is one oxygen and one hydrogen. So this oxygen and hydrogen is covalently bonded, so they're sharing an electron, but this oxygen molecule has such a hefty nucleus that it has the ability to pull one more electron to it as well. So that makes this molecule have an overall negative charge. So we call this a hydroxide ion. So it's a small molecule that is an ion because it has an overall, it has a charge, and in this case it's a negative charge. So it's kind of like, you can kind of imagine that if this had been water, if this had been an oxygen with two hydrogens covalently bonded to it, it's like one of the hydrogens left its electron behind. And so that's how that hydrogen turned into a hydrogen ion. It left its electron with oxygen, which has a bigger, stronger nucleus to create a stronger pull and has the ability to hold that other electron to it. And compared to the amount of pull that a single proton has, the oxygen nucleus can pull more strongly and keep the electron there. So you can kind of think of this as having been a water molecule that's separated apart. And so what we have on one side is a hydrogen ion, so it's oxygen and hydrogen covalently bonded with an extra electron, so it has a negative charge. And then elsewhere we have that lost proton that is the hydrogen ion. Okay, so those two ions are very important when we talk about pH. Now let's go back to thinking about water here for a minute, and thinking about, actually let me go back to, do I have water? Yeah, there's water. So if we think of water in ionic form, or in ionic terms, what that means is we have one hydrogen ion and we have one hydroxide ion. So the number of hydrogen and hydroxide ions is equally balanced, okay, in water. So we could call water water, we could call it H2O, we could call it HOH, or hydrogen hydroxide. So HOH, hydrogen hydroxide, that would be ionic water, and where there's an ionic bond holding it together, one covalent bond and one ionic bond. And that brings us to pH. Okay, so what is pH? So this is the pH scale that we're looking at. Sometimes it is shown horizontally. This one is shown vertically. The middle of the scale is 7. 7 on a pH scale is neutral. It's not acidic and it's not basic or alkaline. What pH actually means is the power of hydrogen. In other words, so that's the P and the H. So the H of pH is hydrogen. The P is power. Or you could think of it as amount or percentage. It's not actually a percentage, but that's a nice way to think of it. But that's what the P and the H stand for in this term pH. So you've heard of pH before. You know that fluids in the body and fluids anywhere could be measured for their pH. So pH is a measurement of how many hydrogen ions are in a solution. So if we're looking at water, just plain old straight up distilled water, we are in the middle of this pH scale. So the pH of distilled water is 7. And so in the middle of the scale, at that neutral point of 7, what that means is there's an equal number of hydrogen ions and hydroxide ions in the solution. So that's what water is a pH of 7, a neutral pH, and an equal number of hydrogen ions and hydroxide ions, making it up. Now, if we were to look now at the acidic end of the scale, each step on that scale, so if we go from 7 to 6 to 5 to 4, each of those jumps is 10 times more acidic than the previous number. So we keep increasing by 10-fold the amount of hydrogen ions in a solution. If we're looking at a solution that's a solution that has a pH of 6, and then we look at a solution that has a pH of 5, that solution that's at the pH of 5 is 10 times more hydrogen ions in it than the solution that is at a pH of 6. So pH is a measurement of how many hydrogen ions are hanging out, wandering around in the solution. So the more acidic we get, the more hydrogen ions there are. So remember, a hydrogen ion is just a proton, and that positively charged proton is going to be attracted to anything with a negative charge. So protons or hydrogen ions can be highly reactive. So if we have a solution that is almost exclusively hydrogen ions, it is going to be a very reactive solution. So anything you put in it, whatever negative charge or whatever electrons are in whatever you would put in that very low pH solution, those hydrogen ions are just going to rip it apart. They're just going to pull those electrons to themselves and completely destroy whatever molecule that was, and they'll like sort of claim all of the electrons as their own. So think of hydrogen ions as being extremely reactive, especially if we have a high concentration of them. Okay, so that is acidic. So the most acidic we could possibly get would be a pH of 0, which is almost essentially a pure concentration of hydrogen ions. Which like if we came into contact with in our skin with something that is like a pH of 2 or a pH of 1, it would burn. Like we would feel it, so meaning that it's causing damage to our tissues. It is pulling electrons out of those atoms that are making up the tissues of our skin, and that's what an acid burn is. Okay, let's look at the other end of the scale. So if we go to the higher numbers that are higher than 7, we're having a negative amount of hydrogen ions in the solution, which is kind of a funny way to think of that we can get more and more negative in the amount of hydrogen ions in the solution. But another way that we can think of it is that the higher the number gets in pH above 7, the more hydroxide ions there are in a solution. So even though pH is always a measurement of how much hydrogen is in a solution, and that's what pH means, you can think of an acidic pH as having a lot of hydrogen ions in the solution, and you can think of an alkaline or a basic pH as something that has a lot of hydroxide ions in it. Now, hydroxide ions could be just as reactive as hydrogen ions can be, but in different ways. So if we had a solution that was almost pure hydroxide ions, and then we put a little something in it, some protons in it or something, same thing, anything with a positive charge is just going to get torn to shreds by all of the negative charge that there is in hydroxide ions. So a strong concentration of hydroxide ions can burn your skin or whatever just as easily as a strong concentration of hydrogen ions. There's going to be different reactions that are happening, but just as damaging to tissue. All right, so let's see if this has made sense so far. An acid is a molecule made up of a hydrogen ion, which is a cation, and any anion. Okay, so if we're calling a molecule an acid, what that means is it is a molecule that is held together through an ionic bond, which means it has a cation and an anion. If we're calling it an acid, the cation aspect of it is a hydrogen ion, and the anion aspect of it is we don't care what it is. As long as it isn't water. If it was water, as long as it isn't hydroxide, if it was hydroxide, we would call it water. But an acid is an ionically bonded molecule that is made up of a hydrogen ion as its cation and anything else as its anion. So hydrogen chloride is an example of an acid. So the hydrogen is the cation, it has a positive charge. The chloride is the anion, it has a negative charge, and they're held together to each other through an ionic bond. Hydrochloric acid is a very, very strong acid. This, of course, is the acid of our stomach acid, but it's a very strong acid, meaning that the hydrogen comes off of the chloride really easily. So the chloride is a big molecule, so it can hold the hydrogen to it, but it has a strong negative charge, and that hydrogen ion has a positive charge. But if you put it in water, that hydrogen is going to come off the chloride, and so basically what what hydrochloric acid does in water is it donates hydrogen ions to that solution, which then is going to change the pH of that solution. So a further definition of an acid. In water, an acid dissociates into hydrogen ions and its anion, donating the hydrogen ions to the solution, making the solution more acidic. So if we go back to our pH scale, if we started with distilled water, which is our pH of 7, and we added hydrochloric acid to it, what we would be doing is donating hydrogen ions to that solution, and there would be chloride ions in there as well, but it's the presence of those hydrogen ions that changes the pH of the solution. And the more hydrochloric acid we add, the more acidic the solution becomes. Now let's look at the flip. Oh, this is hydrochloric acid going into water. So on the beaker on the left, what we see is HCl. That's the formula for hydrochloric acid. We're putting it into the solution, and so you can see there what it's doing is separating. And so the protons, or the hydrogen ions, are attracted to the water molecules, and they turn it into H3O with a positive charge, and instead of H2O, and then the chloride ions are kind of off by themselves. And so we can see that we're changing the solution by putting and so the hydrochloric acid has donated these hydrogen ions to the solution, and has changed the nature of this solution. And so on the right side, we're looking at the exact same thing, but it's just being shown as with like balls showing the atoms rather than the letters. So okay, so this is kind of what's happening when we put an acid into water. We're going to end up with hydrogen ions in the solution that might just float around in the solution, or they might glom onto the water the way that we see it happening here. and change the nature of the solution. Okay, now let's look at the flip side of it. The flip side of it is a base. So a base is a molecule made of a hydroxide ion, and so that's the anion in this molecule, and any cation. So again, a base is a molecule that is held together through an ionic bond. And remember, ionic bonds are not the strongest of the bonds in the world. They are moderately strong. But they're held. But this ionic bond holds together the two parts of a base, and the two parts of a base are a hydroxide ion, that's the anion, so that's the negative charge, and then any cation. So anything with a positive charge can be the cation in a base. So sodium hydroxide is an example. So sodium has a positive charge. If we remember where sodium is on the periodic table of elements, sodium is way over on the left, meaning that it has, in its natural state, it only has one electron in its outer shell. It's happy to get rid of that electron and act as and exist as an ion, in which case it would have a positive charge. When it has that positive charge, it's attracted to hydroxide, which, remember, we looked at hydroxide, which is oxygen and hydrogen covalently bonded, but with one extra electron, so it has an overall negative charge. So the negative charge of hydroxide, the positive charge of sodium, attract them to each other, and they're held together with an ionic bond, and that makes sodium hydroxide. So if we look at this other aspect of the definition, in water, a base dissociates into hydroxide ions and its cation, donating hydroxide ions to the solution, making the solution more basic or more alkaline. We could say it either way. So here we're seeing sodium hydroxide going into the beaker of water, and what it's doing is separating apart. So now we have separate sodium ions and separate hydroxide ions floating around in this solution. And so on the right, we're seeing the same thing, but again, it's just being depicted differently. Okay, so have we changed the pH of the solution? Yes, we have. And so how have we changed it? Essentially by donating hydroxide ions to the solution, because those hydroxide ions are then going to interact with any hydrogen ions that had been in that solution, and they're going to attract them to them. And so in essence, what we're doing is subtracting hydrogen ions out of the solution by adding hydroxide ions. I sure hope this makes sense. Okay, let's keep going. So both acids and bases contain ionic bonds. We've talked about that, although some may also contain covalent bonds. So if we're talking about an ion that is a small molecule, there can be covalent bonds between some of those atoms. But acids and bases, by definition, always also contain ionic bonds, because acids and bases are made out of a combination of cations and anions. And okay, so let's keep going with these definitions here. Okay, so we talked about what an acid is, which is a hydrogen ion with an anion. We talked about what a base is, which is a hydroxide ion with a cation. So what if we had a molecule that was covalently bonded, so we had a cation and an anion, but neither of them were hydrogen or hydroxide? Then what would we have? Wait, more definitions here. In an acid, the cation is a hydrogen ion. Oh, this is just review. In a base, the anion is a hydroxide ion. Okay. An ionically bonded molecule in which the cation is not hydrogen and the anion is not hydroxide is called a salt. Okay, we've heard of salts. These are normal things. We have them on our food all the time, and there are other salts that are not on our food, but a salt is a molecule that's ionically bonded together. The two parts to it are ionically bonded, and there's no hydrogen and there's no hydroxide. Okay, so here's an example. Sodium chloride. Sodium chloride, as you probably know, is table salt. And so what this is showing on the top is sodium and chlorine in their natural states. But we know that the chlorine would like to add an electron to itself, and the sodium would like to get rid of an electron. And so the sodium can donate an electron. Chlorine can take one on, turning it into chloride, which is the chloride ion, and turning sodium into the sodium ion. They now, on the bottom part of the picture, they now both have charges to them. Sodium has a positive charge, it's a cation. Chlorine has a negative charge, it's an anion. Therefore, because they have those charges, they're attracted to each other, and an ionic bond forms between them. So that's what a salt is. Salt is held together through a crystalline structure, and so on the left here, we see these are the atoms, the way they arrange themselves in a crystal of salt. They make a nice regular structure like that. But what happens when we put salt in water? So we had looked at what happens if we put acid in water. If we put an acid in water, we're going to have hydrogen ions donated to the solution. If we put a base in water, we're going to have hydroxide ions donated to the solution, which essentially sucks up any hydrogen ions that had been in the solution. If we put salt in solution, what does it do? This crystalline structure of salt falls apart because the negatively charged chloride ions and the positively charged sodium ions are very interesting to the water molecules. So remember that water, even when it's covalently bonded, is what we call it is what we call a polar covalent bond, or it has what we call polar covalent bonds, which means that the electrons are unequally shared, which means that the electrons tend to hang out, in the case of water, they tend to hang out on the oxygen side of water, so the oxygen side tends to be more negative, and then the hydrogen side tends to be more positive, because we kind of have these protons sticking out there, in the breeze, where the electrons are hanging out more around the oxygen. So we have this slightly positive and slightly negative ends to water molecules. Those ends of water with the different charges get attracted to these the ions that make up salt, and it pulls the salt apart. So water molecules will gather all around. So you can see up on the top right, we see a chloride ion. So it's still an ion, it has its negative charge, but the positively charged end of water, so the proton or the hydrogen ion end of water, is attracted to it. So it gets close to it, and it gets so close to it that it kind of pushes away the sodium. And we can also see these sodium ions here that are attracting the oxygen, the negative end of the water molecules. So what we are looking at here on the right is sodium chloride in water. So that sodium chloride falls apart, it dissolves in water, and that is what we would call salt water, of course, right? This is now not water, with salt in it. The salt has fallen apart and dissolved in the water, and that makes salt water. Okay, so our last little bit that we need to talk about on this video, that has already gone on too long, I apologize for that, is buffers. So buffers are weak acids or weak bases that help to regulate the pH of a solution by absorbing or donating hydrogen ions to the solution. So buffers are important in any system where we need to maintain the proper pH of a solution, which any living system is that, because the pH of a solution is, a solution is where it is the environment within which chemical reactions happen. If there are a bunch of hydrogen ions in that solution that aren't supposed to be there, those hydrogen ions are going to wreak havoc and change what reactions can happen there. Same thing, if there are a bunch of hydroxide ions in there, they're going to pull apart those molecules. They're going to pull hydrogen ions out of other molecules and destroy those molecules, and they're not going to work the same way as they would if they were in a different pH. So it's important in the body for us to maintain proper pH in all of our fluids, and of course not every fluid is kept at the same pH. Blood has a different pH than urine does, which has a different pH than stomach acid does, which has a different pH than pancreatic juice does, which has a different pH than the mouth, and a different pH than our interstitial fluids. So because there are different reactions that need to occur in each of these fluids, we have to keep each of them at the proper pH. Not all at the same pH, but they each have to be kept at their proper pH so that they can function correctly. So buffers are how we do that. Buffers are weak acids or weak bases that are in solutions that can interact with the solution and assure that the correct number of hydrogen or hydroxide ions are in that solution. Because sometimes we do want hydrogen ions in the solution. Like in the case of stomach acid, we want to have an acidic solution there, because those havoc-wreaking protons that we have in stomach acid are what we use to denature proteins. That's the first step of digesting proteins that happens in the stomach. We expose proteins that we're digesting to hydrochloric acid, but that hydrochloric acid essentially makes the pH of the stomach very, very low. So now all of these hydrogen ions are there to interact with the molecules that are there, and they essentially start to rip them apart, and that's exactly what we want to have happening in the stomach. Okay, so again, buffers are weak acids or weak bases that help to regulate the pH of a solution by absorbing or donating hydrogen ions to the solution. So keep in mind that both hydrogen and hydroxide ions are quite reactive, but in different ways, and they both will change what can happen in a solution. And so so on your notes, I don't think I have it here on the slides. No, I don't. In the notes, you'll see at the bottom of the notes, there's an example of a buffer system. So that is on page 4 of your notes. If you look at that, what you see is, so if we look at the right side of it, what we see is carbonic acid. Carbonic acid is a weak acid. So it's made of 2 hydrogen atoms, a carbon atom, and 3 oxygen atoms. So we know that, because I told you that carbonic acid is a weak acid. So that means that if we put carbonic acid in water, some hydrogen ions are going to come off of it. So if we run this equation that we're looking at here on page 4, if we run it to the left, what we see is carbonic acid losing some of its hydrogen ions and turning into what we call bicarbonate ion, which is a weak base. But we can see that it has donated hydrogen ions to the solution that it's in. Excuse me. Okay, so we've added an acid to a solution, and hydrogen ions have come off of there. So we've made that solution. So if this whole reaction is happening in a solution, we've made it more acidic by adding that carbonic acid to it. And the carbonic acid then has turned into bicarbonate ion. Now bicarbonate ion so you can see that there's a double arrow or a double-sided, a two-directional arrow in this equation or in this reaction, which means that the bicarbonate ion can retake that those hydrogen ions back up again, which would then raise the pH of the solution. So let me just say this one little side note here before we wrap this up, that one thing that could be a little confusing when talking about pH is a lower pH is more acidic and a higher pH is less acidic or more alkaline. So, just kind of keep saying that to yourself as we're talking about these things. So we add an acid to a solution, it donates hydrogen ions, it makes it more acidic, which means it raises the pH. So if there were a lot of hydrogen ions in this solution, though, in the solution that had this bicarbonate ion in it that can act as a weak base, some of that bicarbonate ion is going to take on some of those hydrogen ions, and that's going to drive this equation to the right. So the bicarbonate ion will turn back into carbonic acid, and it will pull those. Or in doing so, it will pull some of the hydrogen ions out of the solution. So this is an example of a buffer system, and there's actually more to it than this, but this is a great example of a buffer system where we can see that hydrogen ions can be donated to a solution or they can be pulled out of solution depending on what that solution needs. And with the correct amount of carbonic acid in the solution, that will assure the right amount of hydrogen ions that are free in the solution, and that will assure the proper pH, and that will assure that the chemical reactions that need to happen in that solution can happen the way that they need to. Okay, more on that in the next video, but this concludes the material on exam 1. So let's take a look at our objectives. We looked at, we defined organic and inorganic molecules, and gave examples of each of those, and then we defined acid, we defined base, and we defined salt, and we looked at the pH scale, and hopefully the pH scale makes some sort of sense to us at this point. and we started to talk about buffer systems. So in the next video, we'll be talking about the fluids of the body in which the buffer systems work. So we need to define all of those fluids, and then we'll take a look at some different buffer systems that we have in different fluids of the body that help to maintain proper pH so that the body can work the way that it needs to. So homework following this video is complete exam 1 on Populi. So you don't have specific homework directly for this exact lecture. This just wraps up our exam 1 material, and your homework is just to complete exam 1, and that's due by next class time. So have a good time with that, and then after you complete this, then you can watch the next video, which then sends us into our next round of material. So we'll look at fluids, we'll look at pH balance in the body, and then from there we'll go on to looking at the organic molecules in the body. Okay. Hope this was enjoyable and informative, and I will see you next time in class.